So my name is Phil Bourne. I'm from the University of California, San Diego, and I've worked with Helen for, I guess, over 20 years now. I should say worked for, of course. And I have to say it's been a wonderful experience, and I'm very proud to be here today as my boss. But I also get to, even a greater pleasure, because I get to introduce the previous boss I had some 20-odd years ago, namely Wayne Hendrickson, who's professor at Columbia University. And I think many of you, Ken already took part of my time, so I only had a minute to start with, so I only have a few seconds. And I just want to say that Wayne, I think, is known to probably most people in this audience as an absolutely outstanding structural biologist who's contributed an enormous amount both to the methodology of structure determination, but also lots of very interesting structures that have incredible biological implications. And he also has a third and final achievement that he probably doesn't even realize, and that is that when we sit down at the RCSB PDB and we decide to do things, we come up with these ideas, and Helen will say, "Would Wayne like that idea?" And I'd say, "Probably not." And she'd say, "Well, forget it then." So he doesn't even know he's the gatekeeper of the PDB, RCSB PDB. So Wayne, with that.
Okay. Thank you, Phil, and thanks everyone for letting me be here to celebrate with Helen. Like many of you, I've known Helen for a very long time, but the part that I know her about that I'll discuss today is the little bit that we've done together on structural genomics. And Helen has been a mainstay of the effort on the protein, the structural biology of the structural genomics operation, the protein structure initiative. And I will tell you about our efforts in this line with respect to membrane proteins. But before I do that, just by way of background on structural genomics itself, although it wasn't called that, structural genomics has been with us really from the get-go. This is what I call Kendrew's zoo of myoglobins. You all know about, I think, sperm whale myoglobin, the very first protein structure ever worked out. But that came about because Kendrew, first of all, searched to find organisms that would have myoglobins that would be very good for crystallography. And many of these, as he says, do crystallize, including the myoglobin of the ass. But the one that we know about is the one that crystallized the best. And that's the theme, really, of the efforts that we do at the New York Consortium on Membrane Protein Structure, which is funded by the NIH Protein Structure Initiative. And what we have done that I want to briefly tell you about is to work in the first phase that we were involved in, which was the second phase overall, PSI2, on targets that originated from E. coli with an effort to try to understand globally what the universe of membrane proteins would be like. And currently, in the PSI biology phase that we're in the midst of, we are working from the membrane protein component of the genome of humans. Initially, we worked from E. coli seeds through a process that included NMR as well as crystallography, and currently we're only using crystallography. Otherwise, the pipeline is very much the same. I'm not going to belabor the pipeline at all, except to say that we start out with organisms with a particular gene that exists in the human genome and expand that into organisms for which we have genetic material from which we can clone. And in the case of the bacterial or prokaryotic generally, also archaea, we clone from these various organisms. At the moment, it's more than are on here. It's now about 140 that we are using. And when we work on eukaryotic genes from eukaryotes, then we clone from materials, usually synthesizing the material from these 10 organisms. And the results of these studies have led to a number of structures, and I'm going to tell you about 3 of those today. And I just flash these that came from our first round. The thing that I would like to impress you with is that the work by this sort of search, this attempt at biological discovery using this approach, not necessarily knowing anything about the gene at the beginning, has led to papers that you see are in prominent outlets. That is to say, the results turn out to be pretty darn interesting, and I'll try to convince you for the ones that, some of which are not published, like this one that I'll tell you about, and wherever it was, this one that I will tell you about that is published only in a pre-publication kind of way, not about the protein, and another that's not on here. These are, I think, novel and interesting results. Okay, so I'm going to quickly go through 3 of these topics, one of which is published, and it was on the first of those battery of slides. It's a bacterial homologue of an anion channel that exists in plants. And this is a busy slide. I just really want to show you this graph right here, which is a graph that shows that low CO2 and high CO2, how open the apertures are in the stomata of leaves. This is one stomatum with the 2 cells that define it. These are called guard cells. And by doing a selection based on this cloning with CO2 selection, they were able to identify the protein, a protein that is responsible for the opening of this. In the case of a different experiment, this opening aperture of 3 micrometers across this width would be the case that you would see when light is on. When light is off, it closes. You don't need to do photosynthesis. You don't want to lose water vapor when you're in the dark. And so it's controlling that. And it's done in part by this ion channel that was found by this means. And the ion channel that they found is shown here as the genetic framework. There are 10 transmembrane segments. And it turns out that there's a bacterial homologue, which has actually a misnamed name of TEHA. And we found that TEHA molecule through the process that I was telling you about. It turned out to be a gene that was in a family of 3 families in a superfamily that turns to be the same one as the SLAC1 gene from Arabidopsis thaliani. And here you see the overlay of the structure-based alignment of the TEHA bacterial sequence with the plant sequence down below. The color code here is the color code of the program conserv. The purplish ones are highly conserved. The cyan ones are less conserved. And in fact, every odd helix is highly conserved. There are some other conservations that I won't take time to go through. And that conservation is such that this protein molecule shown here has the odd numbered helices shown in red. The even numbered helices are in green. The sequence is up, down, down. Here's the start, helix 1, helix 2, helix 3, helix 4, et cetera. So it's a pentamer of, if you will, a quasi-5-fold symmetric object that is organized here. And it makes a trimer. And down the middle seems to be the pore. And we've done a lot of work, some of which I'll show you, to indicate that it is a pore. But the pore is blocked by this phenyl group. And the next slide shows that a little better. Here's a picture of the same view that you saw. This is the view from the other side, and the side view with that phenylalanine highlighted. The electrostatic potential surface shows you where the belt is. That's a definition of the membrane boundary. And if we do a cutaway, we see that that pore does exist. You can see it here. And it's blocked by this phenylalanyl side chain. What this pore does is to pass ions, and I will have I don't have time to show you the whole of that. I'll show you one aspect of it. And that aspect is largely about this phenylalanine, which is identically phenylalanine in 812 out of 814 sequences that we carried. So I think the exceptions only prove the rule at some level. The structure that has that phenylalanine is shown here. That's the wild type one. We have mutated the phenylalanine to an alanine and shown that the pore is open. I'll show you those data in just a minute. The mutation, one of the mutations that was selected in those selections was a glycine at this position that's mutated into an aspartic acid residue. We made the analogous mutation in this bacterial protein and found that it was totally blocked, even blocked, when you co-made the alanine mutation that opened the hole. The evidence for the opening was done by us following a recipe that some other people had published just ahead of us, not based on the structure, just making transformations of these it's RNA injections into xenobacillocytes. And I thought my time was already up, but I guess. And this is the picture of, I'll just take you through the injections of the plant protein. And in the wild type case, there are currents. They get enlarged when there's a particular kinase that activates them. I won't take time for that. But if you make the mutation of that guarding phenylalanine to an alanine, the pore becomes wide open. These are the traces at different voltages, and you can see that there are very large currents. Here it's that bar is a 1 microamp. We don't have a lot of poop in this. 1 microamp, and in the next one down, the F450A1, these are the 5 microamp bars. Back to 1 microamp here when we do the double mutation or just the G194D, and both of those are essentially closed. So how does it all work? The one bit about how it works that I will want to tell you about today is, I should have said, the structure is at very high resolution, 1.15 angstrom resolution. So when I show you the picture here that's in the heavy bars for the side chains, it's something we feel we really know. The phenylalanine side chain there is in a conformation that is not ideal. As you may know, many structural biologists here, if I think of my hand as a phenylalanine side chain, C alpha here, C beta at my elbow, C gamma at my wrist, the preferred conformation for the phenyl group is perpendicular, the chi2 angle being plus or minus 90 degrees. And what you see in the actual structure, the heavy bonds, is that it's more like 0. It's right in the worst possible case. A hydrogen atom from the ring is pointing exactly at the hydrogen atom from the C alpha backbone, and we really want to get away from that. In this case, it cannot get away because the leucine here and valine there would have blocked it. It would have been too tight a collision if that were the case. And what we did was to make mutations. Oh, that might be better. Yeah, thank you. What we did was to make mutations of that phenylalanine to several residues, including leucine, valine, and alanine. And any of those mutations have released that, leading to shifts of a 1/2 to 3/4 of an angstrom in the C beta position, and concerted shifts throughout the helix involved. And that in turn leads to then, and enough said, I guess, that in turn leads to the opening of the channel completely in the alanine case, for example, but with the leucine there, it is barely open at all and is now not controllable by the typical kinase activation. Now, where am I? What I'd like to turn to now is the second example that I want to get, and this is give you information about, and this is a work in progress still, and it's a work in progress despite the fact that this structure, which was solved using part of the technology development from the PSI initiative on sulfur SAD. I won't take time to tell about it, but we got this structure about 3 years ago and have been puzzling over that structure for some time. And the reason has been that it appeared to be a symmetric molecule, a tetramer perhaps, or was it a dimer out of that tetramer? We didn't really understand it. I'll show you more about this. This is the view that we thought was the view through the membrane. And this piece right here is a piece that's these pair of molecules coming from that. Was unsatisfactory for various reasons that I'll get to in a minute, and as a result, we've been pressing on to get this. We've just now recently gotten a new structure from a different species of the same cis-C molecule. Here's the electron density distribution for that. It's a pretty modest resolution, and we're still trying to improve everything. But here is the model that comes from that. And what we find is that that model is like the diagonal or transverse dimer that I gave you a picture of before. So here's what we think is going on. This is the picture from the dimer that's shown here in the side view, and it led us to the idea that this was the transmembrane part. It turns out that that's just not right. The reason that it's not right is that we think the truth is this, in fact, and one half of this, there's another partner down here that's making this transmembrane part. What's happened is that these there's a connection between these two that's not ordered, and that connection, that piece, we find out actually in the new structure has been moved into this position. That is to say, it's sufficiently flexible and doesn't have its own necessary integrity in the idiomarina structure that we did first, whereas in this Pseudomonas structure, we get a structure that looks like this, and that the transmembrane portions are in that fashion. I don't have time to give you all of the evidence for that, but the bottom line of it is that we think we have a structure that has an opening here. Our evidence is that it actually acts as a channel, that sulfate ions go through this interstitial part between the helices of this 4 transmembrane helices and several reentrant helices. And I think that by persevering, we obtain this picture. Now, the last example, which I'll go through quickly, is a very recent structure of a protein that comes from a family that is illustrated here. It includes a molecule called Bax inhibitor 1. It has an anti-apoptotic function. These are the human, the families TMBIM 1 through 6 that are indicated here. 1 through 4 are closely related to the YET-J gene that we have done a crystal structure from the bacterium. So these proteins, we believe, are giving us a real clue about cancer functions that are happening in the human genome. These proteins are also called, for the 4 of them, LFG1 through 4, and I'll talk about LFG4 as I go here. Here are the 6 human proteins compared to the bacterial one. I won't go through this really in any detail, except to say that if we do this structure-based alignment that you see here, we find that the 7 transmembrane helices are as we would predict for the entire family. And in fact, there are some highly conserved residues indicated by these asterisks. This aspartic acid residue and this aspartic acid residue are conserved throughout. And for the first 4, there's additionally, an arginine residue at this point that you'll see structurally in just a minute. So the structure was solved again by sulfur SAD experiments in this state at high pH, and another structure was done at low pH with a platinum SAD experiment. The result of all of this is that we get a structure that looks like this. So it's a 7 transmembrane helix protein. 7 TM proteins are things that we've known about for a long time from the GPCR business. This is not at all like a GPCR. Number one, the N and C termini are on opposite sides, N terminus in versus out in the case of the GPCRs. And secondly, the cross-sectional arrangement of the helices is radically different. That is, the helices here essentially go 1, 2, 3, 4, 5, 6, 7 around that. And if anything, there is a centrality of helix 3, importantly. In this case, the last helix, helix 7, is central to the whole structure. The structure has a kind of pseudo-symmetry. That is to say, there's a triple helix sandwich, I call it. Helix 1 coming up from behind, helix 2 coming down, and then helix 3 going between the two in this fashion. That's repeated by a quasi-twofold symmetry that brings 4, 5, and 6 that way, and 7 comes up as if it was a helix going sort of in both directions to complete these sandwiches. So it's a completely novel kind of an arrangement. The structure at the high pH is shown here. The key thing is that helix 2, it makes that structure, it's the one that has the helix at that position for helix 2. If we go to the low pH structure, everything is essentially the same, except that helix 2 is now swung out and the hole, there's a hole left where that was. We'll see more about that hole. And this is the superposition of the two units. So helix 2 is movable. Why does it move? It moves because that then leaves a pore that you can see through here. That's the pore looking from the outside inward. And the pore is largely lined by negative electrostatic potential. In the closed state, that interface is lined by a lot of water molecules and blocked, in this case, by this asparagine residue and this phenylalanine residue. If we then do an experiment, these are on bacterial cells. We've also done a similar experiment on proteoliposomes and found, in fact, there's a pH dependence to this, but the bottom line is just on the on the transfected E. coli cells carrying this Bacillus yet-G gene, we see an uptake of calcium. And that calcium leak, that experiment was actually stimulated by observations on the BI1, human BI1 and human LFG4 molecules that have calcium leak properties of that kind. When we look at the B factor plot, you see that there's a lot of mobility at these joints. They open up and it's even more mobile when it's displaced. These are the electrostatic potential surfaces that I will skip out over right now. And I'll just finish this off by relating to you results of experiments that we have done that are illustrated here. The crystals at high pH diffract reasonably well beyond 2 angstrom spacings and have these unit cell parameters of that kind. We can soak those crystals into pH 6, and when we do, there's a pretty large unit cell change, and the structure goes to the low pH closed conformation form. And then if we can return that to pH 8 and get the same structure back, even though there's a noticeable change in the cell constants. And if we go in between at pH 7, we get a structure that is the superposition of the two. And 55% in the open state, about 45% in the closed state. Namely, the pK of this seems to be in the vicinity of 6.8 or something of that sort. The structure that is done by this soaking medium gets to the same structure as the one that we obtained with this other form of the crystal, this orthorhombic form, and the overall the packing is completely different, but the structures are the same. So there's a pH sensitivity to the opening process, and that pH sensitivity is explained by this unit, which has these two absolutely conserved aspartic acid residues hydrogen bonded to one another, and another arginine residue that is contacted in the case of the closed state. So there's what I call a diaspartyl arginyl latch that holds them together. And what transpires, last slide, is that that there is a PK. We can do calculations of the PKs. We've done an experimental verification of this transition, but the bottom line of the interpretation is that in the closed state, we see what I just showed you. There's this charged pair of the arginine and the aspartic acid residue, meaning to say this really is not protonated. But at lower pHs, this becomes protonated, and the arginine is evicted. The pore opens up. It's now a channel. There's additional evidence that, in fact, that channel can probably bind peptide substrates as well as a target for protein-protein interactions that are important to the anti-apoptotic activity, as is the calcium leak. So what I really want to summarize and say in the context of looking at the people who have worked these experiments is that these three examples are typical of the examples that we have gotten from the effort to do relatives of human proteins that we can make in convenient ways in this structural genomics approach. And all three of the examples that I've given had very little information about them ahead of time, absolutely no structural information, but little biochemistry and biochemical information. All three structures are entirely novel. There's nothing like it in the protein data bank before we do these structures. And the results are sufficiently inspiring that you can do biochemical and cellular experiments that actually get to the bottom line of how these proteins perform the actions that they must perform. So with that, let me stop and say thank you, Helen, for everything you have done for our community. It's phenomenal, and we appreciate it a lot. Thank you.
I was intrigued by the sulfate channel that crosses the membrane on a diagonal, and I was wondering, it seems that to cross on a diagonal you would need a longer dimension of the hydrophobic region than to cross straight through the bilayer. So do the dimensions allow you any predictive capability for other membrane proteins that might be oriented in such a fashion?
Haven't really thought about that, I guess I would say. There are big issues for this and also for the SLAC1 anion channel that I described, in that we don't see any sites. In fact, for none of these three examples, although there are ion channels by their actions, we have not found any of the ions bound to these sites. For the SLAC1, we know a lot about the electrophysiology and the selectivity, and it's consistent that they should be passing ions in such a way that there wouldn't be any binding sites. So that may be what's happening. But that then means, in the case of this sulfate ion channel, that there is a long passageway, longer than if it were doing the shortest possible route, that has to be traversed with some kind of slippery interface. No contacts that are binding, but they need to pass. And so what we find on those surfaces is largely hydrophobic residues and hydrophilic residues that are polar but not charged. They tend to be rich in serines and threonines, for example. And how that works is one of the unsolved puzzles in all of what we have.
Wayne, those are fantastic structures. Okay. I'm curious, so you have some amazing resolution structures, but are these crystallized in micelles or lattice, and how do you think that whatever the milieu is where you get these high resolution crystals may affect, similar to this question, the width of the membrane spanning region, and are you compressing or allowing it to expand based on the
Well, in the cis-Z
milieu?
case, we think it's just a very pliable molecule and that it probably does not have pore-like integrity except in the context of a lipid environment. All of these crystals, all three of the examples, or four or five examples, there are several different crystal structures here. All of these are with detergents, very different kinds of detergents, so I could go into that. For the cis-Z one that's very flappable, we recently have gotten crystals in the pidicubic phase, and we're keen to see, are they now going to be better, more realistic? And we don't even have any diffraction data on them yet, but we're hopeful that we will get some kind of a feeling about that.